Alrighty. Well, hey everybody, good morning and welcome to the Monterey Bay Aquarium and the Monterey Bay Aquarium Research Institute's social media feeds. My name is Patrick. I am right here in the middle. We should be going live right now on Twitch, on Facebook, on Periscope and Twitter, on YouTube. We are everywhere right now. Thank you so much for joining us, everybody. And as you can see, I'm not by myself. I am surrounded on all sides by amazing folks here. We are here to talk a little bit more about the deep sea. So introducing folks to my, let's see, over here to the side, that is Emily, the better half of the social media team here at the aquarium. How you doing there, Emily?
I'm doing pretty well over here across the bay in my house. Everything's going okay. Hello to everyone in the chats right now. I'll be hanging out with you and kind of grabbing some of your questions for our special guests today.
Awesome. Thanks, Emily. And let us know if we're sounding good over there as folks are tuning in. And so then right here on the bottom row of our Zoom, we have the intrepid folks over there at MBARI answering your social media questions over there on MBARI's feeds. So directly below me is George Matsumoto. Over here in the corner is Cassie. If you enjoy the social media, that's Cassie and Susan over here. So thank you so much for joining us. But over here on my right, to this side of the screen, we have our very special guest here this morning. We have Astrid Leitner, who is going to be telling us all about her work in the deep sea here. Astrid, how are you doing today?
I'm doing very well. Thanks for having me.
Awesome. So can you tell us a little bit about what your job is here at the Monterey Bay Aquarium Research Institute? What are you interested in? And then we'll dive in, as it were.
Okay, so I am a postdoctoral research fellow at the Monterey Bay Aquarium Research Institute. So that basically means I just finished my PhD and I'm starting my first kind of their short-term jobs. And so this is a 2-year appointment that I have at MBARI. And I describe myself as a couple of different things. So I'm an ecological oceanographer. I kind of coined that term. That bring together ecology and oceanography. And I'm also a deep sea ecologist, and I'm an ocean explorer. And so here at MBARI, and generally throughout kind of my work so far, I've been interested in how the shape of the seafloor, so that's bathymetry, affects the distribution and abundance of animals. Basically, mostly focusing on deep sea fishes, but I'm branching out as well. So that's kind of what I'm doing right now.
That's awesome. Oh, and I can see here, I didn't have the chat. The chat's on your face right now, Astrid. That's my bad. Sorry, that's a production side thing. But amazing. Yeah, so you're talking about that bathymetry there. Hold on. I took a screenshot. You may have heard that. So you're talking about the bathymetry there, and that's actually something that we can dive into right now to talk a little bit about this deep sea habitat that we have out here in the Monterey Bay and the kind of really cool topography and bathymetry that you have available to you for study. So if I can nail this transition, I think we should be good. Yeah, here we go. So this is an animation to show you a little bit about what we're talking about here for why we're studying the deep sea. That's MBARI there, about 20 miles north of the aquarium, and it's located at the mouth of the Monterey Submarine Canyon. If you were to drain the bay, you would effectively be seeing the Grand Canyon, but underwater here in our backyard. The Grand Canyon of the Monterey Bay, the Monterey Submarine Canyon here, is about a mile deep, so similar to that real Grand Canyon you may have seen out there. But it also has a mile of water stacked on top of it at its deepest point. So you've got 2 miles deep of ocean relatively close to shore here, meaning that scientists like Astrid and the rest of the MBARI folks can go out there with immediate access to the deep sea. And that 2 miles depth there is about the average depth of the global ocean. So you can see way out there at the mouth of the canyon just how deep it is over there. And so that is some bathymetry, some topography of note. And now, if I can nail this transition, nearly so. Hold on. Let's see. Oh, I got to switch back over. There's us now in the corner. Okay, so What you're looking at right there, that is MBARI itself. So it's right there next to Moss Landing Marine Labs, next to the Moss Landing Harbor. If you're driving down Highway 1, you may have seen it on your way over. And here are those iconic smokestacks there in the background of the power plant there in Moss Landing. This is the Rachel Carson, one of the research vessels that MBARI uses. Another one is the ROV Western Flyer, which you may sometimes see out there on the bay. And aboard these vessels are incredible tools known as ROVs, or remotely operated vehicles. This one right here is the Ventana, our window into the deep sea. And this one right here is the ROV Doc Ricketts, which you may have seen behind George on his Zoom background. And these robots go down into the deep sea, piloted by folks there up on the surface. And they're the ones that allow us to bring back these incredible images from the deep. They're equipped with all sorts of tech, but in our case here, what we really focus on is that huge camera there in the middle that you can see there on the ROV Ventana. Those images are then piped directly back up to the surface to these control rooms where scientists like Astrid are able to sit in the relative comfort of the rocking boat there on the surface to watch what's going on in the deep sea for hours on end. So with that, that's a little bit of the background of MBARI. And now if I can nail this transition, we should be back here up on screen. Oh, I'm not doing a good job over here, everybody. Hold on. Where is everybody? Here's everybody. It's been a minute since we've done these this broadcast, so bear with me. Thanks everybody. So anyway, that's a little bit about MBARI. So Astrid, we just saw a little bit of that amazing canyon, that topography out there. Before we hop into questions, I'm sure there are already some there popping up. Before we hop into those questions, can you tell us a little bit about topography, bathymetry, all of those metries out there? Tell us what is that all about?
Sure. So here's and let me see if I can scoot. So here's another kind of 3D rendering of the canyon. And the canyon has really complex kind of sinewy turns and also very steep walls like people saw in the animation. And so I'm focused on looking at these walls because they bring together two really different communities, kind of what we think of the seafloor, more shallow water associated fishes, things, that we might be more familiar with, like rockfishes. They bring those right next to kind of the open ocean pelagic community, right? So I'm interested in looking at how that midwater pelagic community has to interact with the more seafloor benthic community at these really sharp transitions that we see in the canyon, like there, if I can point out in my back. Maybe, yeah. So this is one of our sites here, and it's a ridge that comes out, and we actually use the ROVs to dive on both the kind of open ocean side of that ridge and on the land side of that ridge. And then we have another site, which is right about there, which is another really steep wall. We're talking about almost vertical looking cliffs when we drive up to these. They're really impressive. And we're looking at the animals that we normally think about as being midwater animals, things like hatchet fishes and lantern fishes and krill and surgested shrimps, and all these animals basically being pushed up against this wall where they meet predators on the canyon floor, things like rockfishes. And such.
Yeah. Right now behind you, Astrid, I have a hatchet fish playing there. Remind me the other fish species that you were just describing there. So here's a hatchet fish, really beautiful. Can you describe what a hatchet fish is real quick, and then tell me those other species again so I can pull them up?
So hatchet fishes are these amazing fish. They're really laterally compressed. I'm not sure which clip is playing, but they look like they've almost been smushed completely on the sides. And they are really shiny and reflective on the sides, and on the little bit that they have that's their belly, since they are so compressed, they have these wonderful photophores for counter illumination. And depending on which species we're looking at, they also have these really cool eyes. Some of them have tubular upward looking eyes, some have very round big eyes on the sides. but they're really amazingly adapted to living in the mid-water. The reflective sides are actually a form of camouflage when you live in an environment with mostly downwelling light. And they have these, again, these photophores that, even though they already reduce their shadow as much as possible by being so smushed, the little bit of shadow that they do have, they can actually counter illuminate to make themselves basically disappear entirely, which is really amazing. And they do actually, these vertically migrate as well, which is something that I'm focused on if we want to talk a little bit about vertical migration.
Sure, yeah, we can definitely talk about that. Sorry, Cassie, I'm covering you up with a hatchet fish. I hope that's okay. But here, I'll actually cover you up even more with the hatchet fish. But so the there we go. All right, Cassie's a hatchet fish now. But so yeah, everything that you just mentioned there, by the way, if you folks out there head over to our YouTube page, you can watch more of these mysteries of the deep. Everything you're mentioning there, Astrid, from counter illumination, photophores, deep sea camouflage, reflectivity, all those things, those have all been mentioned over the time. So you start seeing all of those themes popping up there. So thanks for the refresher there. And I love hatchet fish, especially that last little part where it turns towards the screen. But Emily, I'm sure at this point there have already been some questions here for Astrid. I want to make sure to get to those. So what do the folks want to know so far about bathymetry and then some of these first little fish species that we're talking about?
Yeah, always a very popular topic when we talk about things like counter illumination and photophores. Astrid, can you kind of dive into what actually makes those photophores work and what other kinds of animals use counter illumination in the deep sea?
Sure. So I would probably send folks over to Steve Haddock's or time in the live stream. He focuses on bioluminescence. It's really not one of my specialities or areas of expertise at all. But photophores are basically little light organs that a lot of animals, not just these fishes, have, where they can actually produce their own light. And so sometimes these are mediated by bacterial symbionts that actually live inside of those little organs, and sometimes it's the animal itself that's creating it through a biochemical process. But yeah, so these animals can actually control the amount and the brightness oftentimes of that light that they're emitting, and so they can match almost perfectly the amount of light that is downwelling. So people have done experiments with these animals where you basically put them in the lab and change the amount of downwelling light, and they can control the amount of light that's released from their photophores to make sure that they are masking their shadow as perfectly as possible. So these are really incredible organs, a really incredible speciality that these animals have evolved to basically try to disappear and hide their shadows from predators.
So cool. We also had a really wonderful question about those hatchet fish. In particular, we're talking about their eyes. Is the eye difference an indication of a different species of hatchet fish or just one species with multiple variations? People are curious.
Yeah, so there are different groups within the family that is the hatchet fishes, the Sternoptic. I'm pretty bad at pronouncing the family names. I write them out. I don't usually say them. I just call them the hatchet fishes.
I think a lot of us can sympathize with that.
But there are two different groups, and they are distinguishable by these eye morphologies. So there's several species within each group that do have these tubular versus these rounded eyes. But yeah, it's definitely a distinguishing characteristic.
That's excellent. And just for the folks out there who were wondering what was playing in the background, just then for you, Astrid, I was playing a lanternfish with its photophores, its belly lights, very prominent there on the bottom. And what you were mentioning there, bacteria, symbiotic bacteria living there in the fish, helping the fish out with those. So that was playing. And then before we got back on Hatchetfish, I did have a strawberry squid up there for a quick second, because those also have photophores on their body. So it's not just fish. Lots of different animals have those counter illumination lights. But I see here, Astrid, that you have hopped over to a different image in the background. So what's going on back there?
Yeah, so these are actually some fishes that we've caught. I can kind of shimmy back and forth here, that have really amazing photophores. This one is the hatchet fish we were just talking about, but this is an example of one with the tubular eyes. And then over here we have a dragonfish, and you can actually see that this one has different colored photophores. So it has some bright hot pink ones along the jawline, and then under the eye, that bright yellow one. And so they're not always the same color light emissions either. There's some really fantastic adaptations for different colors, and we're not always quite sure what these could be good for. But one idea is that this is for signaling between individuals. And there's another animal, I don't have it up here, but it basically has like a red photophore underneath its eyes. Some people call it the spotlight dragonfish, I think. But that's thought that to basically give it like a sniper scope, because it's a really rare thing to emit red light. And so it might be something that only it can see, and it can use it to basically search the deep sea without being detected itself, because most animals aren't very sensitive to red light, since there is basically absolutely no red light in the deep ocean.
Yeah, and I have that video playing right now behind you there of the shiny loose jaw there with its red photophore there beneath the eye, effectively, like you're saying, night vision goggles for things that are using that red coloration to normally camouflage themselves. But if you show up with a red flashlight, suddenly you're very illuminated there. So that's what's playing there in the background. I love how you're pulling everything together from everything that we've talked about over this whole time here, Astrid. So many cool things going on there in the deep sea. So many concepts always showing up.
You know, the loose jaw is also cool because of its loose jaw, not just that red spotlight, but actually when you catch these, they have no skin on the bottom of their jaw. That's why they're loose jaws. And so that might be super strange for people. Why would you have a jaw with no floor? But basically what it allows them to do is open their jaws all the way up, like 180 degrees, to swallow things that are really, really a lot bigger than they are. And that lack of skin at the inside of their jaw allows them to snap their jaws shut faster, because otherwise you have this huge amount of drag to try to close those jaws shut. So those are actually some of my favorite fishes. They're very cool.
Wow, I didn't even know that. I mean, I knew that there was something loose going on there with the jaw, but that's incredible.
They're really awesome.
Awesome. Okay, well, Emily, what do the folks out there want to know?
We just had a really awesome question pop up here, kind of going back to what we were talking about with those photophores about, and this has nothing to do with the deep sea, Astrid, but people have heard in the news about a new discovery around platypuses glowing. Now, they aren't glowing with photophores, they're biofluorescing. So those are two different things. But if I don't know if you know any more about it. Otherwise, I did want to make
very little about platypuses.
not a common animal in the deep sea. I don't understand, Astrid. Why aren't you a wealth of knowledge about these monotremes?
Fluorescence is definitely different than bioluminescence. Yeah.
Yeah, I just wanted to make sure that we made that.
Just in case, there's any like crossover between deep sea and platypi, just in case. You never know. Weirder things have happened.
They are venomous. I know that.
What was that? They are venomous. Yeah.
They are venomous. Absolutely. But folks, check out the new science coming out about platypuses that biofluoresce. They aren't using photophores, so you can check that out if you want to know the difference between biofluorescing and bioluminescing. Definitely check out that stream that we did previously with Steve Haddock, wealth of knowledge all about that information. That'll be our slight little plug here for folks to go back on the
learn the difference between fluorescence and luminescence.
And I just want to chime in. We also have a video called The Allure of Fluorescence on our YouTube channel that gets into that as well.
There you go. That's Susan with the with the hot tip there for the content. If you want to learn all about light, is a really amazing thing on the planet because it works so differently in the ocean compared to on land, but it's used so much. So, Astrid, that's something you're mentioning those different types of eyes in the deep sea. It's pitch black down there, and some animals go towards, all right, well, eyes not necessary, I'll focus on a nose or like some other type of sensory structure. But then there's also all this competition and all of this struggle between not being found even in the pitch dark, where if you're playing flashlight tag down there, having really well developed eyes and light trickery systems. So it's really incredible. Up here we kind of deal with the light that we have around us. We don't have too many bioluminescent things outside of maybe some fireflies, some mushrooms here or there that glow, but that light in the deep sea, as important as light up here for that survival, right?
Definitely. Yeah. And there's so many different applications we are just discovering that these animals are using light for. It's really incredible.
And I have a question for you now that I have you up full screen. Hot pink and yellow photophores. I need more information. I need more of an explanation here. What's going on?
That is something I actually need to bring up with Steve. I haven't seen that. When we saw that animal, I took that photo actually through the lens of our dissecting scope at sea, and I was like, this is amazing, because I thought that one of the only animals that had multicolored photophores was that loose jaw with the red under the eye photophore. And so I don't know much about this yet. I was really excited to see that. The other amazing thing about both this dragonfish and that dragonfish is you can see how black these animals are. They're so pitch black that it's even hard to just take a photo of them. If I lean this way. So this dragonfish is so dark that we actually had a paper come out from the Midwater Ecology Lab that I'm part of with Bruce Robeson. I wasn't directly involved in the paper, but just looking at this coloration of the intense black pigment that some of these animals have to hide in that darkness. It's really incredible.
Wow. Yeah. That almost reminds me of that. Recently, there was that story about the blackest black paint that came out that absorbs so much light that people were putting on cars or other things and was becoming a hazard because you literally can't see these things coming at you. So that's really interesting as well. So it just paint yourself completely pitch black in the pitch black background can also help out, but for that camouflage. So many different ways to hide in the deep sea.
Definitely. And you were saying some of the fishes or I was thinking of fishes because I always think about fishes, but some of the animals that don't have their eyes basically at all, that they've lost them, right? And so you see that transition as you go deeper and deeper and deeper, where there's really less and less light. You see some of the animals actually in the abyssal plain. For example, one of my favorites is Typhlonus nasus. It's a cusk eel that actually has no eyes at all. It's basically just like a tadpole with two giant nostrils. It's very funny looking, but some of those animals really have very recessive eyes and just basically don't use light at all to get around.
Oh, I thought we had that cusk eel here. We do not, Astrid, but I will play here real quick a giant cusk eel so that the folks know what's going on there. So we got a giant cusk eel hanging out. And let's get to some audience questions, but talking about eels and eel-like animals brings us to one of my favorite things I wanted to ask you about. But with that cusk eel there in the background, Emily, what did the folks want to know out there?
Well, first, Pat, you've raised a point that's very important now. As we travel down to the deep sea, we do have to ask all of these fishes if they are Anish Kapoor or not, to make sure that he isn't trying to steal their black pigment. But we do have folks who are curious.
That's a niche reference, everybody.
That's a niche reference.
Those who got it.
We've been showing a lot of these different deep sea fish up on screen right now, Astrid, and folks have picked up on kind of a common theme throughout them. They have these huge teeth. Can you explain why it's so important for these fish to have such kind of what looks like, scary maws down there in the deep?
Definitely. Yeah. So the teeth is something that are really, really impressive. And if these were like of a larger body size, right, they'd be real monsters to us. Makes you think of, in Finding Nemo when they go down into the deep sea, right, and they get that anglerfish with the giant teeth. So these animals need big teeth because there aren't a lot of potential prey items that they might be coming across, right? So when there's just not a super high abundance of animals in the deep sea. And so when you come across a potential prey item, you want to be able to catch it. And a lot of these teeth actually aren't fangs like you would think of with like a lion. They're not that firm. They're actually more like a basket. And so it's for trapping prey and keeping them in rather than impaling prey necessarily. One of the great examples for giant teeth would be the viperfish. There's species of viperfish that have such large teeth that there's no room in their mouth for it. And so their teeth go outside of their head and all the way basically to the dorsal side, to the back of their head on the outside. So if you have a clip of that, that would be amazing. But that really takes it to a whole nother extreme. But again, they're not really impaling their prey, at least as far as we think. It's more of a trapping mechanism.
That's awesome. Yeah, and behind you right now, Astrid, I do have the Pacific viperfish playing that you were just mentioning. Also in there, if you were wondering, there was a little fang tooth video clip there with some more of those teeth there. But right now behind you, we do have that Pacific viperfish, and you can see, folks, if you're remembering there, the position of those photophores, you can see 2 rows of photophores there on this viperfish, and then you see those teeth that are curled up over the top of the head. So. Nice. Synchronized clip delivery there. Perfect. Okay, so with that question, thank you, Emily, for throwing that out. Thanks for the folks out there. But Astrid, I wanted to ask you a little bit about eels, if you don't mind. Can you tell us a little bit about eels? Because that's what's behind me right now in my Zoom background. I'm very excited for this. Can you tell us a little bit about some research that you just wrapped up?
Yeah, so this is actually research I did at the end of my PhD, and it's part of a larger kind of international effort to look at this region of abyssal ocean called the Clarion Clipperton zone. And so I can pull up a map of where we're talking here in my background, but the Clarion Clipperton zone behind me is basically this huge region between Hawaii and this is Baja, California, and Mexico. And it stretches across this entirely huge region of abyssal ocean. And they're currently planning on doing some mining there, deep sea mining, specifically for manganese nodules. And so we went out there kind of a couple times throughout my PhD. If I move this way to these 3 areas here coming up above my shoulder. And what we were focusing on was seamounts, so submerged under marine mountains, and how those are different than kind of the surrounding plain habitat. And atop one of these seamounts, we found this crazy amount of eels. And it literally, like how you're sitting right now, surrounded by eels, that was our first image that we got back from our baited cameras when we dropped them on the seamount. And we'd never seen anything like that before. So I decided to really look at how unique, how different this really was by doing kind of a huge literature review of everything people have seen on baited cameras in the abyssal ocean worldwide. And it turns out nobody'd ever seen this many fish ever at abyssal depths at bait, or actually at any other type of carrion or like carcass. So it was an amazing finding, total surprise to us.
And I'm playing right now that video that you shared there of those eels at that bait trap. It's really mesmerizing to watch. Can you tell us? I'm sure folks are wondering about how big are those eels? What kind of species? What can you tell us about these fish?
Yes. So these are cutthroat eels. They're in the family Synaphobranchidae. And we caught a few of these specimens in a baited trap that we put together. And the ones we caught ranged between 20 centimeters, so pretty small, to 60 centimeters, wow. Which is a decent size. Few grams, we're talking like in terms of body mass, a few grams to 300 grams. So a good size range there, but nothing humongous.
Yeah.
So in terms of species, it's really hard sometimes to tell species from the camera, especially Synaphobranchids. They have very specific characteristics. Often you need to like open their mouth and look at their teeth and look at their scales under a microscope. But since we did catch some of these, we were able to identify them as being Iliophis arcs. And this is actually a very poorly known species. Looking at fish collections worldwide, there's only been 6 individuals collected. So we doubled that with just our single catch on this seamount, right? So we hardly know anything about these animals at all. And it's pretty exciting. We have donated our samples to the natural history museums to try to get a little bit more information out of these guys, because we know so little about them.
That's amazing. Has that research paper been published yet at this point?
So the paper describing this finding, the observation of all these eels, has been published. It's out. It's actually out on open access as well. So anybody can read it if they're interested. The species description and added kind of information about Iliophis arcs specifically is still in process. I'm not directly involved in that. I'm not a taxonomist, but that will be coming out as well. Yeah. Gotcha.
Awesome. I'm hoping that the title of the paper was "Can't Fight the Eeling," right? Was that considered?
It was Synaphobranchid swarms at seamount summits.
Ooh, we went the alliterative route. I like it. Nice.
My favorite thing about scientific papers are all like the little bits of joy that you can find the scientists that like sneak in there.
Yep.
Absolutely. We try to do our best with acronyms as well.
Nicely done. Cool. Well, Emily, I'm sure folks out there have some questions out there about the eels.
Oh my gosh, there are so many questions out here and so many really wonderful questions. I'm going to try and get to as many of them as I can. But first and foremost, there have been some fights going on in the chat of what is and what is not an eel. Can you make the definitive answer for us here, Astrid?
What is the discussion about what is and what is not an eel?
Oh, what are true eels versus some of the fish that we just kind of call an eel, but they aren't really eels. Okay.
So yeah, especially since we were talking about, for example, cusk eels, which are eel-like, but they're not really eels, right? They're different families. So there's families that are the true eels, the Anguilliformes, and kind of there's lots of different species and genera of eels. Then there's kind of the things that swim with an anguilliform swimming pattern, which is kind of the sinuous, snaky swimming pattern, which are then not true eels, but a lot of people would see them and think of them as eel-like or snake-like, right? So that's a little bit of a distinction, but taxonomically, eels are their own group, basically. Yeah.
And and Astrid, do you know what you call a fish that's been named the eel, and then it finds out that it's not really an eel? Do you know what you call that?
No. What do you call it, Patrick?
You call that an eel pout. And now there's an eel pout up on screen. Oh, it's pouting. Look at it.
Eel pouts are also one of my favorites. I call them sock puppet fishes. Because they're so pouty and they are really, really cute. We actually also in the Clarion Clipperton zone caught several eel pouts that they like to accumulate at bait slowly over time. They're really funny fishes. They kind of army crawl very slowly across the bottom. They're very bad swimmers. So if you kind of scare them up, they'll try to swim up and then they just kind of sink slowly back down. And so they're very slow, but they come to bait. So when we do use our baited cameras, we see them after, you know, almost 12 hours or something, like 10 hours, they finally show up. And then they like to just park there and sit there. And actually, eel pouts held the record prior to this observation we made of eels of the most fishes at bait. And this was after several days. This was in the Arabian Sea. They found like 60 of these guys piled onto a shark carcass.
Wow.
Yeah, so we found 115 of these eels kind of within 24 hours of deploying just a very small bait package. So this was kind of a whole nother, I mean, these eels are really predators. They're opportunistic scavengers, but they're much more actively swimming predators. And you can see that in the video where they're swimming through and really tearing at the bait actively. Whereas these eel pouts, a lot of the time, they do kind of tear at bait, but a lot of times they're actually there to feed on some of the crustaceans, the amphipods that come in to feed on carcasses. So two different modes. But yes, eel pouts also very
eel pouts. Cool. Yeah. Well, I didn't realize that the pun was going to have so much information behind it. So I'm going to pat myself on the back for that. That was a good job on my end.
Also, can we just clarify too, just so everyone knows, that eel pouts are Zoarcidae, that they're the family Zoarcidae, so it's actually not an eel.
Yes,
right.
Exactly.
That Astrid
yeah. Yeah.
Right.
And also in that video clip, if you saw, it was circled up, and that I believe is called donutting or bageling. What's the term where the fish is thought to try to look a little bit more like a jelly with its silhouette that you were mentioning there before, Astrid. The shadow or the shape of the fish can really give it away. And so there are things out there that aren't super tasty, that are round, that are often jellies. And so that's what the thought is there with why it was all curled up there in that ball.
Yeah, and actually more than just eel pouts do that. We see that with midwater snailfishes do that too. And so when we're doing our transects, sometimes it's really hard to tell if your little donut fish is a zoarcid or a liparid, a snailfish. So totally different families, but similar behaviors, similar adaptations
I'm
in their
so glad you said snailfish. I've got the snailfish behind you, the threadfin snailfish right now, Astrid. Look at this beauty, everybody. Get ready if you haven't heard of a snailfish before.
Okay, I raised your snailfish for.
Oh,
what? Oh, okay.
Deepest. That's the deepest one ever seen, right, Astrid?
Right.
Yeah.
So the lab that I was in for my PhD, the deep sea deep fish ecology lab with Professor Drazen, they were, as part of the Hadel program, they deployed this respirometer, and they had just a work camera on it, basically to check to make sure that the respirometer was doing its thing. And they caught this amazing video of what's known now as the ethereal snailfish.
Ethereal snailfish.
Yeah, so that was in the Mariana Trench at over 26,000 feet. So over 8,000 meters. That's the deepest observed fish.
So cool.
Incredible. I'm putting my snailfish, the threadfin snailfish, no slouch in the snailfish department, back up on screen as well. But yeah, that deep sea snailfish there, you folks, deepest vertebrate, right, that we've seen so far?
I know it's the deepest fish. So I maybe
okay.
Vertebrate. Stick with the facts I know for sure.
Yes, no, definitely.
Well, it has to be the deepest vertebrate. There's no other vertebrate. Yeah.
I think so.
It's definitely the deepest fish. So
definitely the deepest.
Super cool. And I just wanted to add that the eel pouts, you were talking about them as like donut fish. The ROV pilots like to call them Cheerio fish.
Cheerio fish. Gotcha.
Right, George? Is there a story there?
All right. Well, Cheerios, I had a pun there about being like cheery and like, oh no, like something scaring them into the circle. Anyway, let's move on because I know Emily, you had some questions earlier that we interrupted.
Yeah, I was just going to make the comparison. You were talking about being able to tell the eel pouts and and the snail fishes, you can't tell one over the other when there are those little donuts in the deep sea. It's just like us, if we're standing far away from a box on the counter, you don't know if there's donuts or bagels inside.
Right.
You have to get up close. You got to find out. Sorry, that was a very bad joke there, but we have some really, really, really wonderful questions.
Donuts and bagels and eel pouts cast the same shadow. You don't know which is which until you get close. Yeah.
They have a tiny little bakery down in the deep sea. They're okay.
That's why they do the vertical migration, is for the rising, right? Okay. Anyway, we're done.
Yeah.
We're done.
Especially over here, yeast of Eden. So anyway, we've got some just kind of basic questions about when you're down there studying the deep sea, Astrid. How do you study these deep sea eels when their habitat is so, naturally so dark? But also we had a question a little bit earlier of why don't we just use red light down in the deep sea with our cameras to study these deep sea animals? Can you talk a little bit about why we use the equipment that we do down there?
Okay, so red light, I'll just talk about that first. There is a kind of trend. We're starting to use red light. It's been hard to use red light in the ocean because red light dissipates so quickly. You can't see very far. And especially when you're studying fish know you're coming, right? They're very sensitive in terms of sounds and vibrations. And so you can't really sneak up on them with a giant ROV very well. And so you want to increase the sight distance that you have. And that's hard with red light because you can only see kind of what's right in front of you. But our lights are getting better now. And so we're trying to develop that technology because you do see we think a little bit less kind of avoidance response with red light. We're still researching that. That's actually a topic of ongoing research at MBARI, for example. But yes, red light could be a really great thing to use. Some of the tools we use to study the deep sea, especially like with these eels, for example, Patrick had mentioned ROVs, remotely operated vehicles, and these are really important tools. Other tools would be AUVs, so autonomous underwater vehicles. We use those in the abyss, for example, to basically, they're like torpedo-shaped robots that just take photos or video of the seafloor. So that could be a very useful tool. We observe these eels using a totally different type of technology. We use landers, so free vehicles, and that's literally, think of like a moon lander, right? We are dropping this instrument package from the ship at the surface, and it sinks by itself, unconnected from the ship. It lands hopefully where you want it to and not dropping it off a cliff or the side of a mountain, which we are worried about when we're doing this kind of research on seamounts. And it lands there on the bottom, and then it by itself runs a program to basically take video, photos, whatever you want it to do, and then you recall that acoustically. It drops a weight and rises back up to the surface, and then you have to find it with your ship and collect it and bring it back and then offload the data. So that's kind of what we used to make these eel observations. And in the abyssal ocean specifically, and very much for fishes, baited cameras are really a standard piece of equipment because, again, these animals are very sensitive to ROVs, lights, sounds, noises, and this baited camera, of course, makes no noise. It does have a light because we need to be able to see, but we turn that light on and off, so we take short video clips, and so we kind of surprise the animals, turn the light on for a little bit, film them, and turn it back off to give them a resting period. And then we also bait it. So it's not just a camera, it actually has bait. So in, I believe, one of the videos that we do have of the eels, you can see them swarming around this weird pole. That's actually our bait pole. And what we do is we tie bait fish, usually mackerel, to the end of that pole. And that creates an odor plume that attracts these scavengers and predators from a larger area. And that means we don't have to run around the deep sea chasing them down. They come to us, right? So we take advantage of kind of this natural behavior that they have to be opportunistic scavengers so that we can film them and observe them.
That's awesome. Yeah. Just then behind you, I was playing some Eye in the Sea footage, which was, is it still out there? The Eye in the Sea?
Now that's a project that it's a really old project, but they, Astrid may not even know about it, but Edie Witter had a special low light camera and red light so they could watch baited and actually see bioluminescence. But that was the ROV filming the Eye in the Sea just so we had a nice shot of it underwater.
Yeah, so there it is over there, folks, on the side, the Eye in the Sea plugged into a really cool cool thing that MBARI has out there called the Mars node, where it's basically internet and power to the deep sea that you can plug a science experiment into. So that was the Eye in the Sea right there, and you can see some of that baited trap there. But then, Astrid, I was also playing your eel video again there in the background, so folks could see what that looked like. Yeah.
I just put this picture in my background of another piece of equipment we use. That's actually the baited trap that we use. That's actually me inside of the baited trap.
No. Is that do people draw straws for that role, or?
So we, it's basically a big lobster pot, but you have to set the acoustic release. So you got to crawl in there and set the acoustic release. So that was what that shot was, me trying to set the acoustic release on the deck of the ship before we throw it overboard. But that's actually what we use to catch and identify those eels that you have in your background.
Awesome stuff. All right. Well, before we move along to another video that we have of some cool stuff that you're working on right now, Emily, what do the folks want to know out there?
Oh, okay. We're going to jump back to dragonfish real quick, if that's okay with you, Astrid, because we had a question coming in here. If you could tell us more about the stalk eye larvae of dragonfish, of the Stomiidae, if I pronounced that right, which I just looked up on the internet. They look like spaghetti noodles with like googly eyes at the ends of these stalks off of their heads. So I definitely recommend folks looking up those stalk eye larvae. But can you tell us a little bit more about them and what the advantage is of having eyes either like at the end of stalks or tubular eyes in the deep sea? What's so cool about those?
So yeah, larvae in general have some crazy forms and crazy adaptations. It's like for fishes as well. Eels are a great example as well. They have these leptocephalid larvae that can get super long and super thin and transparent. I mean, they're amazing. And larvae have to have these adaptations because they're really, really vulnerable, right? They're small and everything can and wants to eat them. And so they have these amazing adaptations for buoyancy, for transparency, to be able to avoid their predators. Specifically in terms of eye stalks, I don't actually know what advantage it gives you to have these eyes way out on the sides, but I mean, it does let you have a movable visual field, so you can do a 360. So maybe it's an adaptation for being able to see more of your surroundings. George is raising his hand. So George,
we saw him come off mute here.
Let's get you in
current
here.
theory is that it enables the fish to pinpoint and find smaller prey using good binocular vision. Because if the eyes are right on the head, they really don't get that ability to find their food.
yeah,
Gotcha.
if you have eyes on the sides of your heads, it's pretty hard to find things that are directly in front of you.
Absolutely. And so just for the folks out there watching, we don't have any video really of those juveniles, but here's a juvenile rat tail, just so you can see what that looks like. And then I'll show you an adult rat tail here, if I can find it in time. Yes.
Rat tails are super amazing. Not only do they have these beautiful juvenile stages with long filamentous projections on their fins, but they are actually some of the major players, dominant players on the abyssal plains, especially here off of California. So rat tails are a really important deep sea predator.
So there's that juvenile rat tail right there. And then here's what the adult rat tail looks like. And then without going too much into it, those pelvic fin rays sticking out there, what would be the role of those, are we thinking?
So maybe it's something that's sensory. Maybe it has to do with buoyancy as well, spreading out your surface area. But yeah, I mean, you'd think, doesn't that make it easier to catch? You know, you get like a piece of that long projection, maybe it makes you easier to catch. There must be some benefit that outweighs that added risk for those projections, but buoyancy might be part of it.
And I also just want to add that, so the rat tail, the adults that you're seeing, or you saw an adult and now you see the juvenile with the cool filaments, we see the adults all the time. They're super common, as Astrid mentioned, but this juvenile clip is the only one we've ever seen in more than 30 years with the ROVs. It's really remarkable.
Awesome. And here is, let's see, here is that adult rat tail again. And yeah, you can see just the difference between the juvenile forms here. But okay, Astrid, we have another video of yours that I wanted to pull up, and I'm going to start playing it here in the background. And you can just tell us a little bit about what's going on there with your work right now. So I've got this video playing now. So tell us what's going on there.
Yeah, so this is, we just got to the end of one of our transects. So I'm interested in what is going on in terms of the midwater community when they run into this wall. And so what we're doing is we're using our ROVs to drive from the midwater, kind of out in the middle, away from the canyon wall, all the way in horizontally until we hit the wall. And so this video was taken at the end of one of these transects when we're right at the canyon wall. So I put behind me as well, we basically start out in the midwater and drive all the way until we hit the wall. And we are seeing really huge differences in the midwater communities in terms of densities and abundances of different animals once we get to the wall, so we're thinking there's kind of this wall accumulation effect. So what you're seeing in the video is at the end of a 150 meter transect. 150 meters is basically where the Monterey Bay Canyon shelf is, where that flat area is before the canyon wall drops off into the deep. And so this is our first horizontal transect that we do. And you're seeing we ran into basically a school of anchovies and baby hake. So those are the little schooling fishes. And on the seafloor, we have awaiting a wall of mouths, basically, of these rock fishes. The large gray ones that you see are Bocaccio. There's also, at some point in the video, a big lingcod that comes through. So the lingcod you think of kind of as a shallow water kelp forest fish. A lot of people might be fishing for lingcod, but they do actually go down pretty deep as well. This is a good example of that. And you also have some other rock fishes there. At some point, I believe in the video, you have a canary that swims through, a canary rockfish. And these are all predators on these smaller schooling fishes, but also on that midwater pelagic community. We're talking, again, shrimps, small fishes, those lanternfishes we were talking about, for example. So by living on that edge, on that canyon wall break, they have access to this kind of accumulation. At least that's what I'm testing for, of animals. And so one of the things we're doing besides using ROV and video techniques is we're also using bioacoustics. And so we're partnering not just with the Midwater Ecology Lab, but also with the Acoustical Ocean Ecology Lab to use sonar to basically map the distributions of animals along the shelf break.
Oh, sorry, we got the lingcod on screen.
Nice.
Okay.
Yes.
And it's gone.
And it's comes back.
Awesome. Well, that's really fascinating stuff, and that's a 10-minute video there. How long do you do those transects? How long do those last again?
So we actually do it by distance and not by time. So we do 200-meter long transects, and we keep that distance constant as we step down the wall. So the transects take us about 6-7 minutes to run, and then once we get to the end, we kind of film and look at the environment that's at that canyon wall, look at kind of the predators that are sitting there waiting for these animals once they get impacted on the wall. And then part of that study too is to try to understand the currents around the wall. And so we have instruments out there that have both a current meter and a camera system on it. And so we're trying to see, are these animals being pushed regularly onto that wall? And is that maybe one of the reasons why the Monterey Bay Canyon is such an incredibly rich ecosystem? You know, krill are big prey items for things, not just rockfishes, but whales. We're talking about blue whales and these incredibly important charismatic animals. And so what we've noticed is that some of these animals tend to feed right on that canyon shelf wall break. And so my question is, are those two things related, right? Are these animals targeting this topographic feature, that break, because those midwater prey items are being accumulated there?
So sort of like having a pantry being created by the currents, where you just head over there. We had a krill swarm there in the background there for a little bit, so the folks can remind themselves of what a krill swarm might look like, what blue whale soup
it really looks like a soup. Yeah, they get amazingly dense.
Yeah. I'm looking at the time. I have no idea how it's 11:57 right now, and how we've been talking to you for over nearly an hour here already. Time sure has flown by. We have two more things that we have for you. And Emily, I know, has been prepping for this. First things first, we have is a speed round of questions to get to from the audience at large. And then we're going to ask you a few questions about how people can get your job. But first of all, speed round. I hope you're ready. Let's see if I can pin Emily next. All right, here. Oh, this looks good. All right, speed round. Take it away. Okay. Oh, you're muted, Emily.
Helps when I unmute myself. Trooper wanted to join the conversation before. So, okay, first, she's going to try and appear in the background now too. First question of our speed round is, can you quickly define for us what is the midwater when we talk about it?
Sure. The midwater is basically the area below the surface ocean, the shallow ocean, where photosynthesis is going on, all the way down until we get just above the bottom. It's all the area in the middle of the ocean where there's no structure.
Excellent. What are fish teeth made out of?
I saw that question in the chat. I don't know if they're made out of the same thing as our human teeth. That is not, I don't know.
All right. We've already stumped her. Excellent. All right. What is your favorite thing, or what do you find the most interesting about studying the deep sea?
I think the exploration aspect. I got into this originally because I was really interested in space and aliens. And then I realized that my chances of seeing aliens in space were probably pretty low. But now working in the deep sea, I see aliens every day. Like, these animals that we're looking at are really alien. And going to some of these places that no human has ever seen before is just so exciting. It really is modern day exploration. And so I think that exploratory aspect is my favorite.
Very awesome. Cool. We do have a very important question. Have you ever seen the Kraken in the deep sea?
I have not seen the Kraken. However, I have seen some pretty awesome cephalopods down in the deep sea. The Dumbo octopus is one of my favorites. We see those even sometimes at bait in the abyssal ocean, and they're just so cute. But the Kraken, not yet. I have not seen one.
Very good answer there. And then this is a question that we like to ask of all of our guests. What got you into the deep sea? And if you could give any advice to a young person, or just anyone in general out there who might be interested in becoming a deep sea scientist, what advice would you give them?
Sure. So I got into ocean sciences first and got into diving and scientific diving actually at UC Santa Cruz through an amazing diving program that they have there. It's actually currently under threat because of COVID. So that is really concerning to me because that really brought me into ocean sciences. And so programs like UCSC's Scuba Slugs program really helped shape my pathway into ocean sciences and the deep sea. But so I started out with scientific diving and kelp forest ecology at UC Santa Cruz, and then got an internship where I was kind of thrown into the deep sea through the NSF REU program, the Research Experience for Undergrads, which was an absolutely fantastic experience for me. That was a summer internship I did in undergraduate, and I worked up at the Hatfield Marine Science Center with two really great mentors, Rick Broder and Waldo Wakefield, and I was analyzing my first ever ROV video with them, and that just kind of opened this whole new world for me. And it was ROV video taken from actually a submarine canyon, Astoria Canyon, and so that got me interested in topography in the deep sea, and that really just set me on to try to go to grad school and complete my PhD. And I'm kind of just continuing my my curiosities, chasing my questions, and I think that's something that keeps me really motivated to stay in academia, is that ability. Like, my job is to ask questions and find answers, and so it's something that's really that's changing all the time. Like, my day-to-day is different. I have to learn new things, new tools all the time, and so it's really exciting. And one of the things I wish I had known earlier is how much computer skills help you in all types of science. So if there's one thing I can tell you, if you're in high school or in college, take some sort of programming or a data analysis class early, because that will totally open up so many more opportunities for you in terms of really doing science and being able to analyze your data. Bring your natural curiosity, but then give yourself those tools that you need to make that into data and science.
That's awesome. Thank you so much for sharing that, all of that, Astrid. And just a final little like anecdote before we sign off here is that Astrid and I both went to UC Santa Cruz together, and we're in that scuba program together back in the day. So go Slugs.
Woo!
We're here. We're everywhere. 2/6 of the stream here. Perfect. Well, with that, everybody, thank you so much for tuning in to this broadcast here with Astrid. Learned so much about all of those amazing fishes, adaptations for the deep sea, topography. I mean, so many different things were brought up here. So definitely, if you're just joining us right now, hit up the replay once we're done here. It'll be available on all of the social media platforms that you're currently watching this on. So don't worry if you're tuning in right now. Once it's processed, you'll have that video available. Just want to say a huge shout out to our panel below of folks answering all of the questions. So great job, Cassie, George, and Susan, for answering all those questions over there. Shout out to you, Emily, on this side of the screen for monitoring all of those questions, for pitching those. Thank you so much for that. And Astrid, thank you so much for your expertise, and wishing you the best with all of the fishes that you're out there looking at. All right. Cool. Well, with that, everybody, thank you so much for tuning in to this Deep Mysteries of the Deep here, all about deep sea fishes and topography. Thank you so much for tuning in. Like and subscribe, follow, all those things. And with that, we will deep see you again soon at the Monterey Bay Aquarium and the Monterey Bay Aquarium Research Institute's social media feeds. Thanks, everybody.
Thanks, everyone.
Woo!